This is exactly right.
Hello, listeners. Before we get to the episode, we want to take a moment to address the June 24th, 2022 Supreme Court decision to overturn Roe v. Wade.
This decision stripped away the right to have a safe and legal abortion. Everyone should have the freedom to decide what's best for themselves and their families, including when it comes to ending a pregnancy.
This decision has dire consequences for individual health and safety and could have harsh repercussions for other landmark decisions. Abortion is healthcare, and restricting access to comprehensive reproductive care, including abortion, threatens the health and independence of all Americans.
You can learn more by visiting choice.crd.co. That's choice.crd.co.
And if you're able to support others, please consider donating to abortion funds.
We encourage you to speak up, take care, and spread the word.
If you need a faster, easier way to get your contact lenses without leaving the house, then let us introduce you to 1-800-CONTACTS.
They've been delivering contact lenses for 27 years and have your contacts in stock, even if you have a strong prescription or astigmatism.
You can easily renew your prescription online with their express exam.
And for new customers, they have extra discounts that you can check out on their site.
I love using 1-800-CONTACTS. I find it super easy to find my right prescription, find my preferred brand, and shipping is super fast. It's fantastic.
Try it. You'll be happy you did.
Order online today at 1-800-CONTACTS.com.
It couldn't be easier.
Order online today at 1-800-CONTACTS.com.
What if you were trafficked into the inner circle of a cult and signed a billion-year contract at 13? What would you do? This Is Actually Happening is a weekly podcast from Wondery that features extraordinary, true stories of life-changing events told by the people who lived them. Follow This Is Actually Happening on Amazon Music or wherever you get your podcasts.
Hi, I'm Umat, and when I was a graduate student, I was doing fieldwork in Panama. It was just this amazing place with tropical birds, and I loved the fieldwork I did. I had to hike up these rivers in the forest and find these insects, and it was so diverse and amazing. But it was also tropical, so we were getting bit by insects. We were getting water in our boots. We were dealing with a lot of animals and things. And there was 5 of us living in this house. It was pretty crowded. 5 of us and a cat. And then one morning I woke up exhausted. I had fever the night before and these awful dreams and a headache. And I knew that something was up. And so I just rested that day, and I tried to kind of, nourish myself, have a lot of water, have a lot of food. And I felt better. By the afternoon, I was able to take a walk and everything. I said, okay, maybe this was just a short-term thing. Everything will be fine. And then that night, it was worse. I had this awful fever, really, really bad headaches. And in the morning, I was just feeling awful. And so, I didn't have a car at the time. And luckily, I got a friend of mine to drive me to a clinic. And so I got to the clinic and I explained my symptoms, and they're like, well, this sounds like dengue, and we're going to test your blood. And so they took a blood test and they didn't find dengue. So they sent me home and told me that, this is probably some virus or something, and you should probably just recover on your own. Just, take lots of water, vitamins, just take care of yourself. And so I said, okay. But the disease just progressed. And so I started getting more fevers. And the next day I was really awful, but it would be also kind of cyclical. So I would feel better and, think it's improving. And then suddenly it would hit me again, and I would get these fevers and headaches and be sensitive to light. So 2 days after I went to the clinic, I woke up and my sister just happened to be visiting. And I was showing her the tropics. I was showing her my field site. And so we woke up in the morning. I was not feeling great, but I was feeling okay. And so we drove up to the Canopy Tower, saw some beautiful tropical birds. And I remember driving back and just suddenly being extremely sensitive to light, slowly feeling this weakness coming over my body, this fever coming. And by the time I got back, I was in bad shape. I kind of hobbled to bed and my head was killing me. And luckily I had a friend who said, I think, we should go right to the hospital right now. This is really bad. And we got to the hospital, and I remember just having a hard time kind of even walking to hospital reception in the emergency room and stuff. And I remember somebody bringing me a wheelchair, and I thought, I was like, I don't need that wheelchair. But as soon as, they brought it to me, I did sit down in it, and I was like, whew, okay, I definitely did need that wheelchair. And I remember my friends talking to the doctors and stuff, and they immediately took a blood test. That was a moment when I knew things were way more serious than I had initially thought. I saw the doctor's expression, and he came out with this paper, and he said, "This is a critical situation. Some of my liver enzymes were off the charts. We need to get to the ICU immediately." So I got immediately admitted to a bed in the intensive care unit. They had me monitored, my heart rate and my blood pressure and everything. And then I was under 24-hour surveillance. Every 20 minutes, somebody would come and check on me, because apparently that's how critical the situation was. So it progressed so quickly, at least from my perspective. And I remember just being extremely uncomfortable and confused. And so I did improve slightly when I was in the intensive care, but it still wasn't good, and they still couldn't figure out what it was. And so I stayed in the hospital, and I just remember there was this routine. Somebody would come and take a lot of blood from me. And then, I would spend my time in my room, and I'd be very uncomfortable. I was still getting these headaches, this fever, but I'd also started getting pain in my stomach. I never experienced this before, but apparently my liver was extremely swollen. And so my stomach had become tight and tender. And it was kind of terrifying thinking that like a single organ has swollen to a level where it's like distending your body a little bit. And I spent almost 2 weeks in that hospital, and they still couldn't figure out what it was. I was not getting any positive tests. They were prescribing me kind of general antibiotics, but eventually I started to feel a little better. I'd lost a lot of weight. I was weak, but after about 2 weeks, I was released from the hospital. But I was still very weak, and it really bothered me that we didn't know what it was. I mean, is this something that was dormant that could come back again and resurge, something this serious? I really wanted to know, could it be, it's just not knowing was really, really stressful. And so I ended up going to Canada. I'm a Canadian citizen, and I went to Canada, and I called the hospital beforehand. And it turns out they had a tropical medicine program going on there with the med students. When I called them and told them my situation, they put me in touch with this doctor who's teaching this course. And it turns out they don't get a lot of tropical disease patients in Canada. So I was talking to him about all the stuff I'd read and try to figure out what I had. And he asked me if it was okay if I could participate in trying to help figure out what was going on. And so he had a bunch of these med students around the bed. And it was like an episode of House or something, one of those medical dramas where they try to figure out what you have. And different medical students gave their hypotheses. The experienced doctor said, well, maybe that's something you should pursue, or, oh, maybe that's not right because, of these chart readings or something. And they were trying to figure out what it might be to try to look for antibodies or some indication of what I had. And so then I left the hospital, and I was recovering slowly. And then it wasn't until 3 months later I got a call, and one of the medical students figured it out. It was leptospirosis. And so when I found out that it was leptospirosis, now I had, a diagnosis. I could start thinking about what the mode of transmission was. And I remembered when I was living in that house with those 5 people and that cat didn't like me very much. And I would go on these runs and on pipeline on this road, this muddy road, and I'd leave my shoes outside. There was this one time this cat peed in my shoes, and I didn't realize until I was walking around, and I could smell something weird. And it was an outdoor cat too, so it was probably grabbing all sorts of rats and rodents outside, and also a potential vector for leptospirosis. So that time I also happened to have this cut in my foot. And so there's this possibility that it was this direct blood transmission that got me infected. But yeah, so that's the story of me getting leptospirosis.
It is. I remember that so vividly, and it was terrifying.
It sounds awful. I remember hearing about it after the fact.
Yeah. Oof. Well, Umat, I'm so glad that you are better and that everything, eventually turned out okay. And also, thank you so much again for sharing your story. Because for those of you who are longtime listeners, you may have recognized Umat's voice from season 1 when he provided the firsthand account for malaria.
Let's hope, Umat, that there's no more diseases that we need to have you provide a firsthand account for.
Let's hope. Hi, I'm Erin Welsh.
And I'm Erin Allman-Updike.
And this Podcast Will Kill You.
And today we're talking about leptospirosis.
Leptospirosis. Yeah.
Can I make a quick confession? I just want to say this off the bat.
Of course.
I don't know why, but leptospirosis, Legionella, and leishmaniasis constantly confused in my brain.
Interesting.
Those 3, and now we've covered all of them.
That's like me and Gerard Butler and Clive Owen. They're the same person.
They actually are the same person, so that's very valid.
Well, and now you don't have to worry about getting them confused anymore because this is just, we're done.
Right.
I am really excited for this episode because when I was going through emails to be like, okay, what do people want to hear? I was so surprised at the number of requests for leptospirosis.
Yeah.
And one of the reasons it seems like is because this is a disease that affects not just humans, but also many animal species. And it's so much more prevalent and important than I think I realized even after, watching Umat go through this horrible ordeal.
Yeah, I think it's easy to think of this as a disease of the tropics or as a disease of, other places, but it's a worldwide pathogen, as we'll talk about. And I think a lot of pet owners and veterinarians are probably a lot more familiar with it than maybe the general public.
Yes, totally.
Yeah. I'm excited. It's going to be good.
Too, for me. First things first.
It's quarantine time.
What are we drinking this week?
We're drinking I Smell a Rat.
And why are we drinking I Smell a Rat?
It turns out, Erin, that rats are a very important part of the overall distribution and prevalence and life cycle of Leptospira.
And I also want to say just right here that we are not trying to intentionally further the stigma against rats.
Don't blame the rat.
Don't blame the rat. Rats are just one piece of the puzzle. Many other animals do this, but the title was too good to pass up.
Yeah, because who doesn't want to drink? Who doesn't want to drink I Smell a Rat? I mean, really.
I mean, once you hear the recipe, though, you're definitely going to want to drink it.
Please tell me, what is in it?
It is a shrub. It's a mango habanero mint shrub. So this is like a drinking vinegar. You muddle all of these, fruits and ingredients together with sugar, let it sit, and then you can use that as a great base for a cocktail like this one with tequila, for instance. Or you can use it as a placibarita as well with no alcohol. Delicious either way. And we will post the full recipe for I Smell a Rat, the Quarantini, as well as the Placibarita on our website, thispodcastwillkillyou.com, as well as on all of our social media channels.
And on our website, thispodcastwillkillyou.com, you can find all of the best website type things you can find.
Do you think people even need to hear this at this point?
I don't know, really. Just check out our website. It's there.
I like that. I like that. Erin, I am so ready to learn. So can we take a quick break and get started?
Let's do it.
If your busy schedule prevents you from doing things you love, like reading, you're going to love Audible.
Audible offers an incredible selection of audiobooks across every genre, from bestsellers and new releases to mysteries and thrillers, business, and so much more.
As an Audible member, you can choose one title a month to keep from their entire catalog, including bestsellers and new releases.
All Audible members get access to a growing selection of audiobooks, Audible originals, and podcasts, and more are added every month.
I love listening to Audible because I can do it wherever I go, when I'm washing dishes, when I'm gardening, when I'm just like sitting and trying to decompress. And right now I am loving this book Hamnet that I've been reading. It's fantastic.
Let Audible help you discover new ways to laugh, be inspired, or be entertained.
New members can try it free for 30 days. Visit audible.com/thispodcast or text this podcast to 500-500.
That's audible.com/thispodcast or text this podcast to 500-500 to try Audible free for 30 days.
Audible.com/thispodcast.
When you're scrolling through social media, how can you tell what's real? Anything can be posted online without being fact-checked, including promises of permanent weight loss.
Tanya Zuckerbrot, the founder of the trendy high-fiber F-Factor diet, had celebrity followers, including Megyn Kelly and supermodel Olivia Culpo. But allegations of troubling side effects with the diet began to surface, and people started to question, is she selling powder or power?
Emily Gellis, a popular fashion influencer, saw these allegations and put the diet on blast to her own large social following. She launched a crusade to expose Tanya and the F-Factor diet. What started as an online feud escalated into the real world, resulting in threats, lawsuits, and a whole lot of drama.
From Wondery comes a new series about wealth, wellness, and influence. Listen to Fed Up on Apple Podcasts, Amazon Music, Spotify, or you can listen early and ad-free by joining Wondery Plus on the Wondery app.
Leptospirosis is a disease caused by a bacterium in the genus Leptospira or Leptospira. These are spirochetes, which means those little twirly corkscrew dudes, like a few other pathogens we've covered, like treponema, the causative agent of syphilis, Borrelia, which causes Lyme disease. Have we done any others?
I was trying to think of that and I can't come up with any more.
Me neither. Those are the two I remember.
Yeah.
So this spirochete, it turns out, is not just one bug. It's not just one species. And it seems like, from what I could read, the classification and species definition seems to be in flux currently.
Yeah.
So different papers have a lot of different numbers cited, but it seems like there's over 60 different species. I think that's the consensus in the genus Leptospira. And importantly, this includes more than 300 serovars. Asterisk, a note, Erin. I would like to talk about this a little bit more.
Okay.
Because in our episode on Salmonella, we talked about the idea of like species and subspecies and serovars. And in that episode, I feel like I did a very bad job of trying to define what serovars actually are. And when it comes to leptospirosis and salmonella and a lot of other bacteria, serovars are really important epidemiologically a lot of times in distinguishing, like, was this outbreak all from this source or from that source, etc. So I wanted to give a little bit of a better definition of what a serovar actually is in this case. Basically, serovars are determined based on the surface antigens, so differences in those surface proteins and things, which, of course, are some of the main things that our bodies use to recognize various pathogens, right?
Right.
And so serovars are like strains or isolates that differ in their surface antigens. In the case of leptospirosis, there are over 60 different species, not all of which are pathogenic, but which include over 300 serovars, at least 200 or over 200 of which are pathogenic.
Mm-hmm. It seems like more are being discovered all the time.
I am not surprised. So anyways, I hope that was at least a little more helpful than my description in the salmonella episode.
It was. It's really interesting.
Yeah.
Before we move back to leptospirosis, I just want to take a moment to share something about salmonella.
Oh, love it.
A listener reached out to us, and you know how in the salmonella episode, we were like, oh, is it pronounced Daniel Salmon or Daniel Salmon?
Yeah.
Person who salmonella is named after?
Uh-huh.
So someone tweeted at us and said it's pronounced salmon because he is one of her ancestors. Which is
awesome.
So thrilling.
Oh, I love that. I feel like we've had a couple times where people have emailed us saying, like, I'm related to one of the people you talked about. And I don't know why it thrills me.
Oh, 100%. Every single time. It's so cool.
Now my heart is beating quickly. I love it.
Back to leptospirosis.
Back to leptospirosis. All right. If you're related to anyone, no. Okay. So anyways, that's the pathogen. A lot of different species, a lot of different serovars. Like I said up top, this is a pathogen that is found worldwide. And while it is more prevalent, or at least more common in the tropics, this is mostly just because it can be an environmentally transmitted pathogen. This is something that can persist in the environment for weeks to months in the water and in the soil, and the climate in the tropics is just a little better suited for Leptospira growth and survival. But this is a bacterium that can infect almost any mammal, pretty much all mammals. Some of them, some mammal species, are very good reservoirs, which means that they can harbor this bacterium, mostly in their kidneys, at really high levels for months at a time and pee it out all over the place, often without showing any symptoms or getting sick at all. Other species are less great reservoirs, and they may get sick to varying degrees. But what's really important is that this can infect literally almost any mammal, including marine mammals, which I always find fascinating.
Yeah. Question. You are saying this bacterium. Do you mean like the genus overall, or are there species differences in terms of like which animals are best reservoirs for which species or serovars?
Great question. Yes, there are differences where some species or some serovars are maybe more common in certain animal species and others are more common in other species. But of the pathogenic species of Leptospira, they can have been shown in the lab to infect nearly any mammal if you expose them to it.
Any given Leptospira pathogenic species.
Right, exactly.
Gotcha.
So I probably shouldn't be saying this bacterium, like this genus.
Okay.
These pathogenic spirochetes. Yeah. But excellent question. So when it comes to humans, we generally get exposed either through direct contact with infected domestic animals like cattle or dogs, or more commonly through exposure to infected water sources or infected soil. And exposure happens in a variety of ways. These spirochetes can corkscrew their way in through any little cuts or abrasions on our skin. They can enter directly. If you get infected water in your eyes or your mouth, they can enter through your mucous membranes. Even if you swallow a bunch of contaminated water or drinking water sources become contaminated, you can get infected that way as well. And that is true for animals as well. So that's the way that all other animals are also being infected, through abrasions on their skin or through their mucous membranes.
Which makes sense about high infection rate in some animals, because if you just imagine A lot of dogs love drinking out of puddles.
As I will, my veterinarian, when my dog was a puppy, was like, oh, we could vaccinate for a Leptospira. Like, is your dog a puddle licker? And I was like, are not all dogs puddle lickers? So yes, if you have a puddle licker, that they will be exposed.
Puddle licker. Unbelievable.
So these highly mobile little spirochetes, once they get into our bodies, corkscrew their way directly into our bloodstream and begin to replicate. They cause a bacteremia, which is bacteria in our blood. That's all that means. Over the course of usually about a week or so. And then once those bacteria reach high enough numbers, and what's fascinating about leptospirosis is that the amount of bacteremia can be incredibly high. Like we're talking millions of bacteria per milliliter of blood, which is very high, even before you show any symptoms. And this is way higher than what we would see from a bacteremia in like an E. coli or something without our bodies freaking out about it.
Why is that? Why does it produce such a high bacteremia?
Well, it seems like our bodies are very good at detecting and mounting a response to very low levels of antigen from something like E. coli, but essentially just don't even recognize leptospirosis antigen until it's at very, very high levels. What?
Interesting.
I know.
Huh.
Right? Then, after they get to these very, very high levels in our blood, these bacteria start burrowing their way through our endothelial cells that line our blood vessels and into various organs. And that is usually the point at which symptoms will tend to start to appear. So the total incubation period is anywhere from 7 to 14 days, could be as long as a month, and rarely is shorter than that.
Okay. And once these bacteria leave the blood and start burrowing and finding their way to organs and whatnot, does that mean that they straight up leave the blood? Would you be able to detect them in the blood?
Excellent question. They don't leave the blood entirely, but these are still difficult bacteria to detect because, like other spirochetes that we've talked about on this podcast, they don't gram stain very well. Mm-hmm. And so they're just not as easy to pick up. And I do think that the amount of bacteremia tends to go down once they enter our organs.
Okay.
Yeah.
Okay. That plays a relevant role in the history section.
Oh, I bet. I can't wait.
Bacteremia. Okay.
Yeah. Okay. All right. Spoilers. Or just foreshadowing.
Foreshadowing. Yeah.
When it comes to animals, various animal species that get infected, they can remain infected and primarily in the proximal tubules of the kidney, which is just so interesting that this pathogen loves that particular part of a kidney for literally months, in some cases up to a year. And then because they're in the tubules of the kidney where your kidney is making urine, this pathogen is just being shed beautifully at very high levels in the urine. So for some animals, this process can be completely asymptomatic. And for other animals, including dogs, and in some cases, including cattle, they can also get sick from leptospirosis. And what's interesting is that especially in the case of dogs, which there's maybe more detailed clinical information, at least that I could find on dogs compared to some other animals, the disease actually can look a lot like the disease in humans. So let's talk about what those symptoms actually look like. Mm-hmm. First of all, this is something that even in humans can be entirely asymptomatic.
You could just be shedding bacteria in your urine. Okay.
And importantly, for humans, some papers call humans dead-end hosts. We're not truly dead-end hosts, but transmission from humans to humans is very, 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 very rare. We can potentially shed Leptospira in our urine, but it tends to be at much lower levels than something like a rat.
Okay, so the general route of infection and like course of infection, where the bacteria go from point A to point B and so on, is the same across these mammalian hosts, but the symptoms that they show is different and the amount that they excrete is different?
Absolutely.
Interesting.
Yep. Mm-hmm. I know.
Okay.
But even in humans, it can be asymptomatic. And I tried to get a handle on how often, and that was very difficult to do. A couple of studies that I read suggested that up to 70% of the time, people tested seropositive for leptospirosis, but couldn't call any febrile disease in the months prior. So it's kind of hard to say. Could it be that someone just is still seropositive, but they had an infection, a couple of years ago that they just don't remember? I don't really know. But we do know that in animals and in humans, it can be asymptomatic. And that's really important when we're trying to control or reduce the prevalence of a disease.
Right.
But when there are symptoms, here's how it usually starts. It starts with a
fever.
Yeah, it sure does. Usually it's a sudden onset of fever along with chills. Usually there's a pretty severe headache that develops and body aches, generalized muscle aches. And the number of pathogens that we've covered, even in this season alone, that start out that exact same way is a lot. So this is something that, especially in the early stages, is a very non-specific disease that can easily be mistaken for a number of other viral or bacterial infections.
Yeah.
Interestingly, with leptospirosis, in contrast to the other spirochetes we've seen, you tend to not have any rashes or skin findings early on. And that's because of the way that lepto goes kind of straight into the bloodstream, rather than having any skin manifestations. Usually, you'll also then start to see GI symptoms like nausea, vomiting, maybe some diarrhea. And in a lot of cases, that might be where this disease ends. So you might get pretty sick, but it'll be self-limited, and then you'll recover. But when this progresses to a severe illness, and this can happen over the course of a few days, and sometimes people get a little better and then have a biphasic illness where then they get better and then start to get worse again. And at that point, whether you just progress immediately or have this biphasic illness, this progression to severe leptospirosis is when we start to see signs of real organ dysfunction. So what does that mean? It means we see signs of liver damage like jaundice, yellowing of the skin, and icterus, yellowing of the eyes. This happens from buildup of bilirubin because of the damage to your liver, as well as hemolytic anemia. So this bacteria can cause breakdown of your red blood cells. That also contributes to the jaundice. A lot of times you'll have dysfunction in various other blood cells as well. So you can have a lot of signs of bleeding because of a decrease in your platelets. So it might be mild, like little purple spots on your skin called petechiae, or can be very severe and have massive gastrointestinal bleeding or pulmonary hemorrhage. So bleeding into your lungs that can be fatal. In fact, if you do see that pulmonary hemorrhage, it's an up to 50% mortality rate at that point. So this is something that can progress to a very, very severe illness.
Okay, question.
Give it to me.
How is it causing all of these different symptoms?
Great question. We'll get there.
Okay. My other question then is about who has these symptoms. Like, are there characteristics that determine or play or seem to play a role in whether someone has an asymptomatic or a mild or a moderate or a severe case of this?
Yeah, that's another great question. The older people are, the more likely that they are to progress to severe disease. I didn't see a ton of data on if the same is true in people who are very young, but I'll talk more in just a little bit about how much specific host factors likely play a role in the severity of infection. And we truly don't know what those specific host factors are at this point.
And do the different serovars and species play a role too?
Potentially, yes. But again, we don't have like great data on like this serovar. There's a couple of serovars. Ictohemorrhagiae, I think that's it, is one that is known to be of like very important human significance because it can cause pretty severe infection. But there's so many serovars and we just don't have a ton of data. So we'll get into a little bit more detail on how it's causing these specific findings. When it comes to the pulmonary hemorrhage, it really is that this bacteria ends up causing damage directly in your lungs that then causes that bleeding to the point where you essentially can drown in your own blood.
How is it causing that damage?
So it's damaging the endothelial cells. It's damaging the lining of our blood vessels.
Okay.
It's reducing our platelets, so it's not allowing for our blood to clot, etc.
Gotcha.
Yeah. So that's the liver and the lungs and blood vessels. But one of the other main target organs of leptospirosis, of course, is the kidneys. And so when this progresses to severe disease, it often results in what's called Weil's disease, which essentially can cause progressive kidney damage to the point of kidney failure. Kidney failure, of course, can be extremely fatal unless you have access to dialysis. And access to dialysis can drastically reduce the mortality rate in the case of severe kidney failure when it comes to leptospirosis. So it's a lot. Mm-hmm. It really can affect all of our organs. It can infect the heart and cause cardiac involvement. It can cause myocarditis. It can cause an aseptic meningitis, so it can get into the CSF or at least cause enough inflammation in our brain and spinal cord. That headache that presented at the beginning can become incredibly severe. But overall, a lot of these symptoms can be really difficult to distinguish from viral hemorrhagic fevers, like dengue fever, for example, or from things like scrub typhus, which we technically haven't covered yet.
No, we have not. But I remember, it comes up again.
Oh, good. Symptoms can last a pretty varied amount of time. When they get severe, they can persist for potentially weeks. And even in the case when symptoms are not that severe, there is some suggestion of like post-leptospirosis type syndrome with things like fatigue, persistent headache, myalgia that can potentially last for months.
If you get leptospirosis and recover, do you have immunity towards it in the future? And is there cross protection with different serovars or different species?
What a great question, Erin. That is such an important question because what it basically, like, if you circle on that question long enough, you ask, can we make a vaccine? Do we have a vaccine, etc.?
Yeah.
You get at least some immunity. How much cross protection there is on different serovars, unclear. How long that immunity might last, again, unclear. There is some data that suggests that like waning immunity over time in people who maybe lived in an endemic area and were exposed as young people and then get exposed again as they're older are susceptible again. So it doesn't seem to be something where you're immune for the rest of your life forever. Mm-hmm. We do mount immunity to it in a way that offers at least some protection against severe infection.
Okay.
But obviously this is a huge range in disease severity. And you kind of already asked the question of like, what is the underlying pathology of this? Why do some people get so sick? Other people don't get so sick. And it boils down to three major things, which we've already touched on, but I'll go in a little bit more detail. One is those virulence factors of the particular Leptospira species or serovar that you get exposed to. So some that are more or less host-specific might cause disease in certain animals and not in others. And some with particular virulence factors on them might be more or less virulent than others. The second factor is the inoculum. So like how much pee-contaminated water did you guzzle down? Or like how many spirochetes did you end up with initially?
Right.
And the third is my favorite, the most difficult, the host factors. Mm-hmm. It's a very nebulous category. In the case of leptospirosis, what it seems to boil down to is our cytokine-related immune response. So cytokines are pro-inflammatory agents, right? These are proteins and like stuff in our body that we make and secrete in response to an infection in order to get the rest of our immune system on high alert to go look for and eliminate this problematic spirochete. But as we've talked about time and again on this podcast, if our body goes on too high of an alert, if we get too intense about this inflammation, then we can end up with tissue damage that's not even directly due to the pathogen, but due to these inflammatory cytokines and all of the other immune response that they stir up. So while we don't know a lot of the details on this, we think that that's a huge part of the pathogenesis, especially of severe leptospirosis, is this intense inflammatory and cytokine response that is generated. And that's what's causing a lot of these severe manifestations. That's what's causing the damage to our lungs. That's what's causing the damage to our liver. That's what's destroying our red blood cells and our platelets and causing us to bleed to death.
Okay. Interesting.
Yeah.
Seems like there's a lot more work to be done in that field.
Absolutely. We don't have enough data just yet to say for sure what specific cytokines are being upregulated or causing the most damage when it comes to this very severe versus not so very severe infection. But I will link to a couple papers that have more detail on what we do know about particular cytokines.
Question about diagnosis.
Oh, great.
So like you mentioned, this disease can be confused with many other diseases. It has kind of these nonspecific symptoms. And what would make someone suspect leptospirosis, number one, and number two, you said it's difficult to test for because it doesn't show up very well on gram stains. How do you then test for it? What are the samples you would take?
Beautiful questions, Erin. So how you would suspect it would be, of course, if someone is having these symptoms that I described, like these laboratory abnormalities that we see, jaundice, signs of kidney failure, but also if they have a history of something that would make us think they've had an exposure to potentially contaminated environmental sources. If they've been whitewater rafting, if they've been jumping off of cliffs into freshwater sources, or if they work in a field like a rice paddy or something like that. So occupational exposures or recreational exposures, that might make us think, maybe this is leptospirosis. It might be higher on your list to think about in areas where leptospirosis is more common and less likely that you'll think about it in places where it's less common, but it's important that it exists everywhere. So probably overlooked in some places. How you diagnose it can be really tricky. So it's hard to diagnose on culture alone, but that is like the gold standard. It is possible to do. It's just more tricky. So there's PCR tests that you can use, but those, of course, aren't accessible everywhere across the globe because they're difficult and expensive. So it's mostly based on serologic testing. So looking for antigens or looking for antibodies against leptospirosis.
Okay.
Yeah. And there is treatment, which is the best news. A number of different antibiotics work, but especially in severe cases, it's also really important to have access to supportive care, like potentially emergent dialysis if someone progresses all the way to kidney failure and things like that.
In terms of antibiotics, you said that the first stage was this extreme replication in the bloodstream, just tons and tons of bacteria. And then that's asymptomatic.
Yeah.
And then when they move into the organs is when people would tend to probably go seek treatment. Does the massive die-off of bacteria at that point like hurt anything about you and your organs?
Oh, what a good question. That's like a thing that can happen with a lot of other spirochete diseases. Not that I read.
Okay.
Yeah, but that's a really interesting question, especially for spirochetes.
Okay.
Because they are still replicating the whole time. They don't just like stop replicating once they make it into our organs.
Okay,
They
yeah,
just
of course.
keep on going.
Just, chugging away.
keep on trucking on. So that's kind of the disease in humans. Because this is a pathogen, like I said, that can infect essentially any mammal, it does cause slightly different disease in different animal species, but the spectrum, especially in things like dogs, can tend to be pretty similar. So there can be asymptomatic infection, and then there can be this severe disease that often still has fevers, jaundice, nausea, vomiting, liver failure, kidney failure. So it's very interesting to me how it tends to affect a lot of the same organs, and then a lot of what you see might be very similar.
Yeah.
That's leptospirosis.
Alrighty.
So tell me, Erin, where did this thing come from? How did we figure it out?
Yeah, I will do my best right after this break. I found it really interesting to read about the history of leptospirosis because I feel like although this is not the flashiest of diseases that we've covered, it hasn't caused these widespread epidemics and left destruction in its wake. Just like something like smallpox or plague, for instance.
Right.
But the story of leptospirosis is a classic tale of observation and discovery. It's really sort of highlights the golden age of germ theory. And I think that what surprised me most about the history of leptospirosis was not how we learned about the transmission cycle or the causative agent of leptospirosis, but how much things haven't really changed. But I guess I'm getting ahead of things, so let's start at the beginning.
Okay.
Leptospirosis has existed for millennia, and it seems to have been globally distributed for quite some time. So that's about as precise an answer as you're going to get for where did this thing come from. That's about as precise as I could find.
Okay, then.
Yeah. And part of that is because of how many different Leptospira species and serovars there are that can cause infection in humans and other animals. And so it's hard to say when Leptospira emerged or when it evolved and where it evolved.
Okay.
And it's also hard to say when humans were first exposed and which particular species or serovars were responsible, because those are also things that would have likely varied geographically. And maybe those serovars aren't even around anymore. And in the scientific literature discussing the history of leptospirosis, generally speaking, researchers refer more to the disease leptospirosis rather than a particular pathogen.
Yeah, I found that really interesting.
I found that interesting too, and I think that just speaks to the importance, well, I think it speaks to two things, really. One is the importance of the group of pathogenic species rather than any one individually, at least historically. And I think the other thing is that maybe it's just kind of speaks to the fact that we don't maybe know that much about the different serovars and their impacts. I don't know.
And they're just so hard to culture. I think they're a difficult bug to work with.
It seems like it, yeah. And that's, of course, not to say that the differences among Leptospira species or serovars isn't important medically or in terms of public health. Of course, like I'm always saying on the podcast, tracing the evolutionary relationships among these bacteria can greatly help in understanding things like transmission routes or reservoir species or disease-causing abilities. Information that, like you talked about, Erin, for some or even many Leptospira species isn't that well known.
Yeah.
In general, the pathogenic Leptospira are thought to have evolved from non-pathogenic species that just live in the environment, which some Leptospira species still do.
They sure do.
And historically, that's how this group of bacteria was divided, along lines of, is it pathogenic or is it not pathogenic? And that is in like the classification. I think that that division is still really important in terms of public health and medicine, but I'm talking about like classification systems.
Right.
But more recently, that division into those two groups turned into three groups, essentially saprophytic, meaning living on decaying material and non-pathogenic, intermediate in terms of pathogenicity, and pathogenic. And some researchers are now arguing that this isn't the best way to classify Leptospira because it just takes into account one characteristic of these bacteria rather than looking at, I don't know, their entire genomes, for instance.
Right. Yeah.
But as far as I can tell, that's still under discussion and probably won't be resolved
by the time
this episode comes out. Yeah, certainly not.
Yeah, it was really interesting how much it seemed to still be in flux.
Yeah.
The whole like classification of Leptospira.
Oh, absolutely. And not just the classification, but just learning more about the ecology and which species are playing a role. There are new serovars that are constantly being discovered. Which species can cause disease? That's still kind of being learned. Oh, that one's just an environmental bacteria. Oh, wait a second. Actually. Yeah. The bottom line of all of this seems to be that Leptospira is diverse. Many different species and serovars can cause disease. And there are so many research opportunities for understanding more about these pathogens from a public health perspective. So if anyone needs a master's or PhD project, go for it. No.
Yeah, do it. I love it. We support you.
But that's just the bottom line for like the ecology evolution of these bacteria. What about the human history part of it?
Yeah.
Given how widespread Leptospira are and how many of them can cause disease in humans and other animals, it's likely, of course, that leptospirosis has affected humans for millennia, as long as humans have been humans. Although there's an asterisk there. I didn't read anything specifically about the historical distributions of these pathogens. I'm not sure if it's known. But one paper I read suggested that an epidemic that occurred around 1616 to 1619 in the area that's now eastern Massachusetts could have been leptospirosis. It hit the Native American populations living in the area particularly hard, with the death toll estimated to be as low as 1/3 of the local population to as high as 90%.
Whoa.
And since the Europeans living in the area weren't as badly affected, some researchers have suggested that rats on the ships introduced lepto to the region.
Got it.
But also, there's like a huge laundry list of other diseases that it could have been.
Right.
And we may never know what it actually was. So lepto is one of the suggested options. As with many other diseases that we've covered on the podcast, changing patterns in human settlement or any other practices that would have increased contact with rodents, especially rats, would have made catching the disease more likely. And if you look through historical medical literature, there are many mentions of things that could be leptospirosis or have retrospectively been decided to be leptospirosis. Like you said, Erin, jaundice is, of course, one of the primary symptoms of leptospirosis, and references to jaundice go back thousands of years. Whether or not that jaundice was caused as a result of leptospirosis is anyone's guess.
Yeah, there's so many things that can damage your liver.
There is this passage, though, in the Hippocratic text that some people believe may be about lepto. Quote, "When jaundice supervenes in fevers before the 7th day, it is a bad symptom, unless there be watery discharges from the bowels."
Interesting.
Yeah.
Huh.
I don't know. I mean, I'm sure that lepto was around. So.
Yeah. How interesting.
Yeah. And medical texts from ancient China discuss diseases referred to as rice field jaundice or rice harvest jaundice. And the descriptions of those diseases seem very similar to leptospirosis. And also old Japanese medical texts describe lepto-like diseases called autumn fever or 7-day fever. In Europe and Australia, there were diseases known as cane cutter's disease or swineherd's disease and Schlammfieber, mud fever.
Oh, okay.
I doubt I pronounced that remotely correctly. And all of these terms were used before anyone figured out what caused these diseases or whether they were even related.
Right.
We should have some sort of jingle for when I mention Napoleon's name, because here he is again.
Oh, I can't wait. Dee-dee-dee-dee-doo.
Yeah.
Here he comes on his horse.
There we go. Oh, Napoleon. His infamous troops in Cairo, Egypt, during the early 1800s may have experienced an outbreak of leptospirosis, which they called jaundice fever. Also, I have to shout out our chlamydia episode where I talked about trachoma and Napoleon's troops in Egypt. And I just, at this point, need to make a list of diseases that these poor troops were subjected to during their time under Napoleon. We should have just done a series on diseases of Napoleon.
Yeah, just where all we do is list them because it would take 2 hours.
But even before germ theory was a thing, what would be later known as leptospirosis had recognizable patterns, and you can tell that from some of these early nicknames. So one was that it was commonly associated with certain occupations, and that these occupations, and hence the occurrence of this disease, were often associated with wet, marshy areas. They were kind of geographically restricted. And this tight link, especially between certain areas or certain times of year and this disease, made it like a shoo-in for miasma theory, which was this pre-germ theory idea that proposed that diseases and disease outbreaks were the result of bad air. And it's easy to see how that would have been a reasonable explanation for leptospirosis, because it did occur in certain areas and it did occur in certain professions, and it didn't really occur in large epidemics the way something like influenza or smallpox did. And importantly, avoiding those areas or removing the source of the bad air, like draining water around mines, it decreased the incidence of the disease. So even before people knew that cane cutter's disease or swineherd's disease or rice harvest disease were the same things, people were able to limit their exposure to it.
That's amazing.
So interesting. Yeah.
Yeah. I love it when those kinds of things happen in these episodes, Erin.
Me too. But leptospirosis wasn't alone in causing jaundice and a fever and occurring in certain marshy environments. There was also yellow fever, infectious hepatitis, louse-borne relapsing fever, and it wasn't until the late 1880s that leptospirosis, or at least one manifestation of it, was officially distinguished from the rest of these possible causes of infectious jaundice. In 1886, a German physician named Adolf Wheal described a disease that involved jaundice alongside splenomegaly, renal dysfunction, conjunctivitis, and skin rashes. He called it infectious jaundice. His characterization of this disease stuck, but the name, obviously, did not. A couple years after his paper was published, another researcher named it Wheal's disease. And like happens so often, giving this disease a name, giving it a description, increased awareness of it substantially. And soon papers were being published describing outbreaks of the disease in coal miners or sugarcane cutters, association with rainfall or swimming in certain areas. But even though people were able to recognize patterns in where and when this disease occurred and in whom, without a causative agent identified, those were just patterns without a mechanistic explanation. And you were also a little bit limited in your ability to do anything. For that satisfying understanding of how all the pieces eventually fit together and whether the cases of Wheal's disease in England were caused by the same pathogen as those in Japan, for instance, we needed a causative agent. And to get that, we have to wait almost 30 years after the clinical description of Wheal's disease.
Wow.
Yeah. So 1886 was Wheal's disease. And 1915, I believe, was when leptospirosis was identified, was when Leptospira was identified.
Oh, my goodness.
Yeah. So why was there so long a wait? And, you know, none of the papers I read really went into that great detail about why, but I think it could have been many different things. So even though this period, the last decades of the 1800s and the first few decades of the 1900s, was this golden age of germ theory and pathogen discovery, that doesn't mean that all diseases received equal attention.
Right.
I mean, if you think about it, again, leptospirosis is not a big name marquee disease, which isn't to say it doesn't deserve that attention. But at the time, there were things like plague or syphilis or tuberculosis that people were trying desperately to figure out because of the death toll, because the number of people that were infected with those diseases. Leptospirosis definitely attracted interest, but it maybe didn't get the focused attention that some of these flashier diseases did. But one thing was working in lepto's favor, which is that spirochetes, the kind of spiral-shaped bacterium that causes lepto, these had already been discovered and recognized to cause disease, the most important of which at the time was syphilis. And so people had spirochetes on their radar, and they should have maybe been able to pluck these little guys out as the cause of leptospirosis, except for this quirk that you mentioned. Spirochetes don't stain well. So they're really difficult to isolate. They can be difficult to culture. And what the eventual discoverers of Leptospira noticed was that, well, they blamed it on this anyway, is that after this huge influx of bacteria in the bloodstream, those levels drop. And they were like, if you don't catch it then it's going to be really difficult to isolate leptospirs.
No, that's not unlike like malaria or something, where you
Exactly.
have a lot of difference in even just like times of day on when you have a high enough burden in the bloodstream to be able to pick it up.
Exactly. Yeah. And
Yeah.
I think that's what they said. They were like, yeah, we didn't see it in the bloodstream. And so if you test a blood sample of someone who's super duper sick,
Right.
especially considering the microbiology technology that was around in the 1910s.
And I want to say, because like I mentioned how like something like E. coli might be at low numbers, you can grow those other bacteria out in culture from blood. So even if you only have a tiny bit of it, you can grow it to be more. But that's really difficult to do with Leptospira. So if you can't see it right away,
Yep.
it's a lot harder.
Yeah. Yeah. That's a really important point too.
Yeah.
So how did people eventually discover what was causing Weil's disease? Seems super difficult.
Yeah.
They went looking for it specifically. It wasn't just one of those serendipitous discoveries. A couple of researchers in Japan named Ryokichi Inada and Yutaka Ito had an interest in Weil's disease for years. And an outbreak of the disease among coal miners around 1914 to 1915 led them to run some experiments to see if they could isolate a pathogen from an infected person. And I also want to point out that their interest was not purely scientific, not like, oh, here's this curious little disease. Let's see if we can find a new pathogen. At the time, mortality rates of Weil's disease at their clinic averaged 32%,
Oh my.
which is incredibly high.
That's really terrifyingly high.
Exactly. And so they wanted to do whatever they could to try to control the outbreaks by finding the source. They looked for bacteria in blood, urine, feces, you name it, in people who had Weil's disease. And when they didn't find anything, they took some blood from these infected individuals and injected it into monkeys, rabbits, rats, guinea pigs. And the guinea pig turned out to be the unlucky or lucky one, depending on your perspective. It developed many of the symptoms that were characteristic of Weil's disease, and when they dissected the animal's liver, they found it to be teeming with spirochetes, which they named Spirocheta icterohemorrhagiae.
Yep. Okay.
Yep. Later, of course, Leptospira. Almost immediately after they published their findings in 1915, researchers in Germany also isolated Leptospira from infected individuals. But the real first-placed finisher, even though he didn't know it, may have been A.M. Stimson, who observed spirochetes in the blood of someone that had been diagnosed with yellow fever. He figured that what he saw, these little squiggly bacteria that looked like question marks, hence the name he gave them, Spirocheta interrogans, was causing yellow fever. For a long time, people were like, it must be a spirochete that's causing yellow fever. It wasn't, as we know. But in retrospect, he did end up being the first to describe a bacterium that later researchers would link to leptospirosis.
Interesting.
The similarity in symptoms between leptospirosis and yellow fever, and not to mention their association with wet, marshy areas, also made diagnosis sometimes confusing. And that could have potentially contributed to the delay in identifying both the causative agent of Weil's disease and the yellow fever virus. Anyway, soon after Inada and Ito and colleagues identified the causative agent of Weil's disease, they next set their sights on uncovering everything they possibly could about this pathogen. And they did an hugely impressive job. From culturing it in the lab, to characterizing the timeline of infection, early vaccination studies, to understanding the transmission route of the pathogen. What they did was set a really strong foundation for understanding the disease and for growing future research, especially research that was focused on prevention. Because as we know, if you want to stop a disease from spreading, you at the very minimum have to know how it spreads in the first place. Mm-hmm. Which for leptospirosis, by the time of the causative agent's discovery, was still under debate. They knew it was in wet marshy areas and associated with water. So could it be mosquito-borne, like yellow fever? Several people seem to think so. And it was a reasonable explanation given the geographic and climate variables associated with outbreaks. And some researchers even successfully carried out experiments with mosquitoes transmitting leptospirosis to lab animals.
Huh.
But it was likely that the mosquitoes were acting as mechanical vectors, like just depositing infected blood, clinging to the mouthparts into the animal. Mm-hmm, mm-hmm. Weil himself thought it was something that you ingested, while others thought vertebrate animals played a large role. Inada and Ito and colleagues demonstrated that the leptospirs could be transmitted through the healthy, unbroken skin of a guinea pig.
Yeah.
Though broken skin was more reliable. I had no idea it could be through unbroken skin.
It can be, yeah. Usually it's broken skin, but especially if you're submerged in, exposed for a long period of time, and especially with wet skin really reduces your skin's, increases permeability, essentially. So then, yeah, it can be unbroken skin.
Wow. Okay.
Sorry.
Yeah.
Mm-hmm.
Yeah.
Mm-hmm. Yeah.
Well, they also showed that it could cause infection through ingesting it.
Yeah.
But where did it come from?
Yeah.
They got the idea to look at rodent kidneys and urine after a different team of researchers observed spirochetes in the kidneys of field mice while working on scrub typhus.
Oh, cool.
Yeah. And so they were like, you know what? All right, fine. Let's gather all of the rodents that we possibly can, house and wild rats, and then look in their kidneys, and boom. They found a bunch of their spirochete, spirocheta at the time, ictohemorrhagiae. And they noticed that the leptospirs seemed restricted to the kidneys and that the rats seemed in excellent health.
Until they took out their kidneys.
Prior to their dissection.
Yeah.
And that pointed towards their role as reservoirs. And this didn't actually surprise too many people, possibly because, A, rats have been maligned for centuries, but also during trench warfare in World War I, cases of Weil's disease shot up, particularly in areas with high rat populations.
Okay. Yeah. So they were like, I suspected it.
Oh yeah, of course it's the rat, which is sad. Poor rats. Rats are great, by the way. Anyway. All of this clinical and epidemiological and ecological knowledge about leptospirosis, which is a lot. I mean, that happened in a very short time span, just a few years. And that's pretty remarkable. And even though antibiotics weren't yet available to help treat the infection, understanding the transmission cycle was enough to enact control measures in some places. And throughout the following decades, into the mid-20th century and beyond, people continued to find out more about this group of pathogens, like how long it can survive in water and under what conditions, what pH is best, what temperature, which animals play a role in the transmission of the disease in certain areas, how disease progression or severity could be tied to certain species or serovars, and also linking all of these previously separate diseases or separate name diseases into one big picture name, leptospirosis.
Yeah.
And another big development was understanding that other animals may not just be asymptomatic carriers or reservoirs, but that many of them suffered severe infection from these bacteria as well, like domestic cattle, pigs, and dogs. The early history of leptospirosis may not be as flashy as some of the diseases or topics that we've covered on this podcast. And there don't really seem to be, at least that I could find, any major fireworks discoveries during the rest of the 20th century. But that doesn't mean at all that this disease is not hugely important. Because despite how much we've learned about this group of pathogens and despite having effective treatment and knowledge of prevention, it is still a huge problem globally, as you're about to talk about, Erin. And since it has such close ties to things like weather and temperature, we can expect to see big shifts as a result of climate change and land use change. So, Erin, can you tell me where we stand with these pathogens of One Health importance today?
I would love to. Let's take a quick break and I'll get into it. Erin, I know that you know and have very recently felt just how difficult it is to get numbers on some of these topics that we cover. And I'm always still somehow shocked at just how difficult it can be. And that was the case for leptospirosis. Very, very hard to get an estimate of what the true burden of disease is, as well as looking at the distribution of that burden, because of course it's not equal across the globe. So this is a pathogen that is worldwide, everywhere except like the Arctic, where everything is frozen. But it is most common in the tropics, and it's very common both in rural areas associated with things like agricultural or like mining exposure, like you talked a lot about, but also in urban, especially poor urban areas where you have lack of access to sanitation and potentially high rat burdens. But to look at actual numbers, if you look at the World Health Organization website, you won't really find any numbers because they don't have any fact sheets on leptospirosis. All of their information is from like 2003.
2000, it's almost 20 years ago.
Yeah. And now there's not a page. The links to like World Health Organization Lepto, like it goes to page 404, not found.
Interesting.
I know. I was shooketh, as they say. PAHO, which is the Pan-American Health Organization, so for the Americas, has some information. The CDC has a page on it. There are probably a lot of other country-specific public health agencies that have data, but it's hard to go through every single country. So there are two numbers that I found, which are wildly different as to global estimates of leptospirosis. Some papers and the PAHO website estimate about 300 to 500,000 cases of leptospirosis globally every year.
Which is a huge number.
Yeah, well, a paper from 2015 that was trying to estimate the global prevalence as well as mortality from leptospirosis. Granted, this is from 2015, so it's a little bit outdated, but they estimated 1 million cases annually and just under 60,000 deaths every year. And
that's a lot.
It's a lot. And this was really just an estimate of severe cases that are bad enough that people are getting very sick and ending up in the hospital. And again, a lot of times this can be asymptomatic. So how many cases are there? Potentially even more.
Yeah. Wow.
Yeah. And there is a lot of data to suggest that the overall burden of leptospirosis is likely going to continue to rise because of shifts in things like climate change that might worsen storms and flooding events, which are really high risk for leptospirosis outbreaks.
Yep.
as well as, like you said, land use changes that might increase urbanization in a way that also puts people at increased exposure to rats and contaminated water sources, etc. So it's not like good news looking forward, especially with how little data we have to begin with.
I think I'm still surprised at this.
Same. I was actually shocked because a lot of papers say, like, this is on the scale of things like schistosomiasis, of things like leishmaniasis, of things like cholera. And I feel like we don't think of it in that way. I think a lot of people think of leptospirosis as a disease of animals.
Or as like recreational exposure.
Yeah, exactly. Which it is. That's totally true. But
Absolutely.
It's also just a global, serious disease. When it comes to animal infections, by the way, it's even harder to get a sense of what the magnitude of the problem is worldwide, as you can imagine.
Yeah.
There are a lot of really interesting papers that look at the overall diversity of animal species that have been shown to be infected. I mean, it really just boils down to like all of them. Like if you try it, they will infect. But it's really hard to get a handle on like what percentage of domestic dogs would test positive, or like how many cattle every year are getting sick from this? How much milk production is lost because cattle get sick? We just don't have that kind of information. I did find one paper that was looking in the United States at hotspots of leptospirosis, like within the US. And one thing that stuck out to me from that paper is that, at least from data from the early 2000s, it did seem like seroprevalence in dogs was increasing, which is not great.
Why is it increasing?
I have no idea.
And there's a vaccine available for dogs as well.
Great question. Yes, there's a vaccine. There's a vaccine for humans too, and for animals, including dogs. Most of the vaccines, as far as I can tell, all of the vaccines that are currently available are whole killed spirochetes of some of the more common serovars, depending on what area you're in. So you asked very early on, Erin, like how much cross protection do you get? Like what kind of an immune response are we talking about? And because the vaccines that exist are just for particular serovars, they're not universal. So they don't cover every given Leptospira serovar.
Seems like a great candidate for an mRNA vaccine, huh?
Oh, yeah. There's a lot of research, mostly on recombinant vaccines, actually, but an mRNA one could be interesting. But recombinant vaccines would allow you to put a whole bunch of different antigens, essentially, antigen components on like one little vaccine, and then give that so you could get immunity a whole bunch of different serovars.
And who gets vaccinated against leptospirosis?
Such a great question. I had a really hard time figuring that out. In the US, definitely dogs, like my puddle licker, is vaccinated. So domestic animals, livestock can get vaccinated. In terms of humans, I think it's relatively uncommon, except perhaps during outbreaks or maybe in occupational settings in particularly high-risk environments. It might be that people are vaccinated. But in general, I didn't even know that there was a human vaccine. I don't think it's very common. Yeah. But that is what I know about the status of leptospirosis worldwide.
It's such an interesting... I truly cannot believe how overlooked this is. I can't get over it.
I really felt similarly about this. Yeah, I want there to be so much more information.
Yeah.
Yeah. Well, sources?
Sources.
Okay.
I have several I'm going to shout out, two in particular. One is a 2015 book called Leptospira and Leptospirosis, which the editor of that book is Ben Adler. And then there was a paper from 2001 by Kobayashi, and that is titled Discovery of the Causative Organism of Weil's Disease, Historical View.
I had a lot of different papers, a couple of actually book chapters that I wanted to shout out. 2 different chapters, one on leptospirosis in humans and one on animal leptospirosis, both from the book Leptospira and Leptospirosis from 2015. So those were great chapters. And then I had a lot on the genetic diversity and serovar diversity of Leptospira, and at least some data on the epidemiology so you can read for yourself. We post all of our sources from this episode and all of our episodes on our website, thispodcastwillkillyou.com.
We sure do. Thank you so much again to Umat for sharing your story again. Thank you also to Bloodmobile for providing the music for this episode and all of our episodes. Thank you to the Exactly Right Network.
And thank you to you, listeners. We liked doing this episode. I hope that you really enjoyed it. Thanks for listening.
Yeah. And a special thank you, as always, to our wonderful, generous patrons. We appreciate you so very much.
So much.
Okay. Well, until next time, wash your hands.
You filthy animals.
Listen, follow, leave us a review on Amazon Music, Apple Podcasts, or wherever you get your podcasts. And don't forget, you can listen to new episodes 1 week early on Amazon Music or early and ad-free by subscribing to Wondery Plus in the Wondery app. You can support This Podcast Will Kill You by filling out a survey at Wondery.com/survey.